genome webinars. I'm Ben Buckus, managing. Hello, everyone, and welcome to Genome Webinars. I'm Ben Buckus, managing editor at GenomeWeb, and I'll be your moderator today. Today's webinar is entitled Translating Big Data into Therapies, Diagnostics, and New Insights into Disease. The sponsor of this webinar is Abbott Informatics. Our presenters today are Atul Butte, Director of the Institute for Computational Health Sciences and Professor of Pediatrics at the University of California, San Francisco, and Executive Director for Clinical Informatics at the University of California Health Sciences and Services, and Randy Heiss, Manager of Global Strategy for Abbott Informatics. You may type in a question at any time during the webinar. You can do this through the control panel, which appears on the right side of your screen. Click on the Q&A box in the upper right side of the control panel. When you click on Send To, please select All Panelists. We will ask our panelists questions after the presentations have concluded. We will now start the webinar with Atul Butte of UCSF. Dr. Butte, please go ahead with your presentation.
Great, thank you very much. And I'm just going to pull up the slides here. Great, thank you for joining us this morning here in the Bay Area. I just want to confirm that you can all see my title slide here. Okay. Let's start then. So thanks for having me this afternoon or this evening or this morning, depending when you're connecting from. I'm really pleased to be able to talk about a recent story that came out of my lab here at UCSF, how we were able to use big data to reach an idea for a diagnostic, then how we were able to use samples tied to an EHR and actually develop that diagnostic into a commercial entity. So I get the next 20 minutes to kind of share this with you. First, obviously, I'm a medical doctor presenting to a mostly medical audience. I got to start with my conflicts of interest, just a few to mention. Suffice it to say that I started a bunch of companies. I consult for a bunch of companies. Some people don't want to believe another word I say over the next 20 minutes. It's obviously up to you, but obviously I'm a full-time academic, but in my lab we do believe that if you want to change the world, you can't just keep writing papers about it. I'm particularly proud of the bottom right on this slide, which are all the companies started by my students. And more than half my students now start companies, even if they go into academia. And they do this with the most amazing platform in the world. That's data and samples. And when you have data and when you have samples, it's amazing what you can do in terms of coming up with new therapies and new diagnostics. So I'm going to show you how they do it and how I do this as well. If you haven't heard, we're in the middle of this data deluge. My favorite example of this is The Economist magazine from 2011. The cover has this guy collecting ones and zeros. This is the issue where they announced that the human species now generates 2 zettabytes of data every year, 2 zettabytes. Now, if you forget your metric prefixes, it goes kilo, mega, giga, terabyte, petabyte, exabyte, zettabyte. And of course, the following year, it's 4 zettabytes of data because this number keeps doubling every 1 or 2 or 3 years. And I'll be the first to admit, most of these zettabytes of videos are just these YouTube videos of kittens playing with pianos and yarn and, entertainment value, but probably no scientific value there in the zettabytes. But there is some the scientific data there. As an example, the Large Hadron Collider, it's my favorite example, in Europe generated petabytes of data for those scientists to develop, to discover their subatomic particles. And obviously where I'm going with this is that we have an enormous amount of data in life science research as well. We have it due to devices like this. Now, I could have shown anything on this slide. I always show this Affymetrix gene chip because this is how I got my start. I actually have no connection to Affymetrix, except my first papers involved maybe 60 of these chips way back in 1997, 1998. Probably the first of these chips to come off the line, looking at cancer cell lines. And now I could have shown a DNA sequencer. I could have shown a mass spec. I could have shown anything in terms of life science measurements, modern measurement technologies. But I think most of the attendees know that we have a lot of these high-throughput measurements now. In fact, when we get tired of measuring them one by one, like on the left, we can measure them even in a 96-well plate format on the right. And everyone runs these microarrays now. It's not a big deal. In fact, we've moved on to RNA-seq and more modern technologies. But what's happened is that everyone in academia starts to write papers about this. And when you write a paper, now it starts with the top-tier journals. It's spread to even more journals. That if you want that paper reviewed, you have to share that data, the raw data, with the public in these public repositories, because the scientists have to double-check things now. And of course, the public should be able to get to those samples as well. So this idea of sharing samples, sharing digital data goes way back to something called GenBank. Probably most people have heard of GenBank. It's the next door neighbor to PubMed, which everyone uses probably on this line. And GenBank is, I think, 38 years old now. It goes back to the first DNA papers. Nobody wants to submit, no one wants to retype those A, D, G's, T's, and C's. So you got to share the data. And so that's what happens in every field of biomedicine. Now, once you have enough people using it, you got to start to share. And so for this technology, for microarrays, for RNA measurements, this paper came out in August of 2012 when we hit the milestone of a million samples publicly available. Now, if you look at this graph really carefully, this was in Nature. You can look at the link there at your leisure. It kind of looks sad. It looks like it's slowing down there on the trend line there. And then you realize they only counted half the year, right? This came out in August of 2012. We're still doubling every 3 to 4 years. In fact, we're just a couple microarrays shy of 2 million samples publicly available, all that data publicly available. So what does that mean? So that means any scientist now that wants to start a project, let's say looking at cancer or diabetes, any scientist can start with the data first, come up with a hypothesis, and then test that in samples, to test that in mouse models. But you can start with the data first. Actually, forget about our scientists, even a high school student, right, who wants to do a science fair project, even she can go to these websites and amazingly search, here I'm showing you Array Express and Geo, that's a European one and the NIH one, and even a high school kid can go to these websites, type in breast cancer, as an example, and find and search for and find 59,000 samples of breast cancer about as easily as you can find a song on iTunes today. That's how easy it is. What an amazing time to actually be a kid doing science fair projects. Right? I mean, you're starting with almost 60,000 samples. Now, if you ever figure out what's so magical about 60,000 samples already run, it's probably more samples than any breast cancer researcher would ever have in their lab, because every one of them has to run these samples, has to digitize them, has to put them into the repository if they want their papers reviewed. Actually, the number I like even better above there is 2445. You can see it there on the left, series 2445. Let's call that 2400 for a second. So what that means is 2400 research groups, 2400 labs have contributed data into this repository. Let me tell you what a ridiculous number that is. Let's pretend I'm a brand new researcher in breast cancer. I'm a newbie. Let's pretend I know nothing. And let's pretend I'm writing a breast cancer NIH grant. And the grant I'm writing is that I'm going to get 2,400 of the best breast cancer labs to run their best characterized patients using the latest measurement technologies, and I propose they're all going to give me that data for free. Now, if I actually wrote that in a grant, you would probably all, if you were reviewing my grant, you'd probably laugh me out of the room. Why would 2,400 labs give you any data for free? Yet here it is, right? Here are 2,400 labs giving you data for free. So when I think of starting projects with big data like this, I actually call it retroactive crowdsourcing. And what do I mean by that? So this is 2,400 labs on the internet there to help you, so that's crowdsourcing. But these 2,400 labs don't even know they've been helping you. Actually, these 2,400 labs are there to help you, and you don't even know they're helping you. And again, if 60,000 samples aren't enough to get started, just wait a year or two, it'll be 100,000 samples. So this is a digital repository of data, just sitting there waiting to get started on projects. Not enough scientists out there to even use this data. Now, if you have any doubts a high school kid can do it, I have to show you the one high school kid that actually did this. This is Brittany Wenger from Sarasota, Florida. I've yet to meet her. She's on the left picture there. She's on the right holding the white trophy. She won the top prize in the Google Science Fair competition because she built an artificial brain, neural networks, to diagnose breast cancer. And she did this with immunohistochemistry data, immunohistopath data, qPCR data. She built a diagnostic with machine learning classifier. I guess she was bored because the following year she did it all over again for leukemia, because you could see she was in the paper. And what I mean by this is not just to kind of showcase a high school kid. I've actually still yet to meet her. But what that means is that all of us can now start to learn to actually start experiments using data first. Start with data, come up with a hypothesis, and then start to go chase it down into samples and mouse models. And that's what I'm going to show you here. So that's what high school kids do with this data. Obviously, where we're trying to get to is precision medicine. Of course, everyone's talking about precision medicine. Now, of course, when the president mentions precision medicine more than a year ago, everyone's thinking about it. And one way to think about precision medicine is in terms of diagnostics. Can we get to better ways to tell when patients have a disease? Not just do they have a disease, what subtype of a disease might they have? Might they have a subtype that responds better to specific therapeutics or not? So that, in that way, there could be new ways to think about diagnostics here that we might not know about what kind of diseases a patient might have or not. Now, my first big grant when I was way back at Stanford, this Marina helped me with the slide, is go get every human disease. You know, why breast cancer, why diabetes? Go get every disease. And you can actually find all of them just by searching for samples with disease samples and healthy samples. So we started to just collect experiments, other people's experiments, publicly available, where they've already run them on microarrays or RNA-seq. And you could just see here, let's pretend every box here is a gene, and the colors represent how much of that gene is there, up and higher and lower. And we can calculate these signatures. And what are we going to do with these signatures? Well, one of the things we want to do is make diagnostics out of them. Now, for every one disease, though, we don't have just one person's experiment. We might have dozens, or if not thousands. We found thousands, for example, with breast cancer, as I showed you. And so what we can do is figure out what's in common across all these experiments. What's the core common set of genes that everyone sees is upregulated? And we start to chase that down to make diagnostics, specifically protein diagnostics. So here's the central dogma here, just to remind the biologists in the audience here, if they need any reminding, DNA leads to RNA leads to protein. The kind of simple thinking here is RNA, obviously we can measure with these microarrays and RNA-seq. That's what we have these great repositories for. But protein, to me, makes the best kind of diagnostics. I love protein diagnostics. The pediatrician in me remembers way back that if I had a screaming child with a fever and a sore throat, somehow I could stick this Q-tip back there and I'd get enough antigen to pick up strep throat, with a few drops. That's 1970s proteomics. That's called an ELISA. If you have the samples and an ELISA kit or these monoclonal antibodies antibodies raised against the antigen, an ELISA is a super sensitive test if you know what to build them against. So I'm going to use the RNA data to try to figure out what ELISAs and what protein tests to go chase down. And we did this for a bunch of these. I'm not going to spend any time on this. We did this for cancer markers to help the surgeons figure out in the middle of a surgery what's medulloblastoma and what's regular brain to help them save as much brain as possible in a brain surgery for a kid. And then we did a transplant rejection biomarker. This is when organs get transplanted in. Can we actually figure out if the body is rejecting that organ? Sometimes the body wakes up and thinks, this organ isn't part of me anymore. I got to get rid of this. So the body starts to reject that. That's called acute rejection. It's not painful. Patients don't know this is happening. So we wanted to come up with a universal blood test for transplant rejection. Universal means I don't care if a heart went in or a kidney or a lung transplant. If it's being rejected, let's see if we can find a diagnostic for it. So we had a couple of these kind of papers. The one I'll spend a little bit of time talking about is this disease called preeclampsia. Now, a lot of people have heard of preeclampsia, but a lot of people have not. This is when the blood pressure goes shooting high during pregnancy. This disease kind of falls right between the cracks. The obstetricians take care of pregnant women, of course. And, I love them, but they sometimes say that the delivery is the cure. If you just take the baby out, the mother gets better from this shooting high blood pressure. You've got to treat them with mag sulfate for a couple days, but the mother gets better. But we pediatricians then have to deal with these tiny babies, keeping them alive for months, because there's really no choice here. You have to take the baby out because obstetricians have to take care of that patient first. So this disease falls right between the cracks. It's still how husbands lose wives around the world. I think there's 4 drugs still in trials for preeclampsia, but our diagnostic is still kind of ancient. We don't really have that modern of a diagnostic. We're using urine protein, general urine protein, which is pretty nonspecific, especially in today's obese Americans. Not hard to really use that as a diagnostic. So we wanted to come up with a new diagnostic here. So Linda Liu, Matt Cooper, and Bruce Lin got involved. Here, let's start with Linda, searching for data sets. So if you just search the repositories for preterm birth and preeclampsia, again, we're going to start with the data. You can see just how many experiments are out there using preterm birth preeclampsia. 299 in the first one, 249, 154, 143. So many experiments with already the data sets run. These are digital repositories. We download the best of these, and we start to do a kind of experiment. So we start with NCBI-GEO. That's this repository, the Gene Expression Omnibus. From the genomics, we could figure out how many samples and do a meta-analysis. We could do some protein work with some samples we had in our repository linked to patients. We can intersect those. We have 13 from the informatics, 9 from serum proteomics, and 11 overlapping that we want to chase down here. So again, we're starting with digital data at the top, a sample bank linked to an EHR at the bottom, and we got at least a way to intersect using public data and our own private data to figure out what might be in common. We're not going to trust any one of these, but figure out what's in common across all of these experimental modalities. So of the 11 to chase down, what are you going to do next? Well, we got to go chase these in new samples. And a new way to do this, what really changed my life as a researcher, is how amazingly organized now there are sample banks and repositories all around the world. So for here, what we did is we ran out of samples, really. I mean, we had some already that we could use in our own research group, but to really run a larger study, we actually contracted with a company called ProMedics. So ProMedics actually organizes human clinical samples. This is all IRB approved. The patients have consented, but companies come in to now organize these samples from medical centers that actually don't have medical schools, that don't have academics, that take these samples and start to organize them. So here we were able to just literally purchase these samples for research use and have them sent to our core facility in our proteomics lab to actually get these protein candidates actually tested. And so here you can see ProMedics, there's a spring 2011 biorepository update, so we can get more than 2,000 pregnancy samples, so we get to pick and choose, which stage of pregnancy do we want, how many samples from each, and then we can order ELISA kits to actually chase down our proteomics. So the experimental design, we have control pregnant women and preeclampsia, early gestational age and later, 16 in each category. Again, we can purchase all of these. We have our 11 candidate serum proteins. We chase them all down using ELISA kits as a crude way to get started with proteomics again, and we were able to qualify 6 of them as serum proteins that we indeed could validate in this experimental design. And here's an example of one of them. This is hemopexin. The pink represents elevation. The green represents the normal pregnant women, the pink are the preeclampsia women, and you can see regardless of gestational age, but this is statistically significantly higher. So we had 5 others like this. It's funded by March of Dimes and an internal seed grant at Stanford. And you can see the link here to chase down the papers and get more details about this. And finally, the last few minutes, what do you do next in Silicon Valley? You got to start a company on this, right? If you want to change the world, we got the grants, we got the papers, you got to get this to patients. And so there's nothing wrong with starting a company out of academia to do this. And to make a long story short, after getting all the preliminary data to work, we were able to spin out a company and raise $2 million in seed financing. Now, to be clear, this mostly academic attendee audience here, I'm not showing you $2 million to brag about. I'm showing you $2 million because I think that's a new way science will continue out of academia. Okay, we got the grants, we got the papers. If we want to get this to patients, we got to take the experiment further to launch a prospective trial with these types of findings. So we licensed that into a startup company, Carmenta. As you can see on the bottom right here, I'm most proud of this one because we already sold this company. It's already been acquired by another company called Progenity. So start to finish, from getting the data to having the unmet need to developing it to testing the samples off the internet, our own local samples, launching the company, selling the company, start to finish, 24 months. Of course, we can do more of these, and I'm giving away all my secrets because we have so much unmet need in biology. But we couldn't have done this if we didn't have samples ready to go to validate these types of findings. The computational findings by themselves are still just not enough to pursue. They have to get tested and move on from there. Now, of course, I'm a big fan of my own samples, and we're seeing a lot of academics actually, including us at UCSF, trying to build out our sample repositories. I'm just showing you 4 here of different academic groups and companies organizing electronic records now. I think samples are best organized when they're linked to electronic health records. I think that's a no-brainer for most people in this audience. I'm just showing you Geisinger here that's been organizing their medical records for 20 years, now helping other hospitals do that with a startup company. Explorers is a company that came out of Cleveland Clinic that IBM acquired last year. Explorers had their medical records and claims data out of Cleveland Clinic. The one that's covered there is Pittsburgh Pittsburgh launching their own medical records, clinical data assets with a $100 million investment. And then just a couple weeks ago, IBM bought Proven Health for $2.6 billion, another organization of electronic health records. So IBM's doing this with Watson. And it's amazingly potent when it's linked to the actual sample. So Geisinger Regeneron at the top here, their deal has been public for at least 2 years. Regeneron sequencing Geisinger patients. It's an amazing collaboration, from my opinion. 23andMe has their samples and their patient records. Now, these aren't electronic medical records, but they've raised another $100 million because they have that organized. Illumina and Helix are down there in the bottom left there, raising $100 million, getting more patient data. And Human Longevity now launching in Southern California, I think they're trying to get 1 million patients now genome sequenced with medical records and medical data. So the cohorts of patients, the samples are best organized, most potent when they're linked to electronic health records. And that organization is hard. That's why you need LIMS systems. That's why you need an organizational system to actually put the most respect you can on that linkage between samples and electronic health records. Okay, I'm going to close it out by just mentioning all my collaborators here for all my work. This is not just this project, but I have to always show my collaborators. I never get any work done without them. And I always have to thank UCSF for endowment support. My lab has a significant number of grants, 17 NIH grants, I think, over the past couple of years from all those different institutes of NIH, March of Dimes, JDRF, HP, HHMI, stem cell money, disease-specific foundations. To thank everyone else who supports my lab, the admin and tech staff, and all the folks who've been mentoring me and helping me along the way. So with that, I will turn it over to questions, I think. I think we do questions next time. I've already seen a couple here posted here. So let me give that back to you guys.
Thank you, Dr. Bu. We will ask questions to panelists after both presentations have concluded. As a reminder to attendees, you can type in a question for the panelists at any time during the webinar by clicking on the Q&A box in the upper right side of the control panel. We'll now turn the webinar over to Randy Heiss of Abbott Informatics. Please go ahead, Randy.
Hello, everyone. I'll just spend a few minutes talking about and adding to what Dr. Butte was speaking about and talking about the very large volumes of data and how to handle them and how to organize them and how Abbott Informatics has helped along that line. We'll talk a little bit about what some customers, providers of biobanking, people that are linked into this business are looking at in terms of organizing this information. One of the problems is that there are a few key messages that we can take away from the bioinformatics field and the challenges that are facing it. One in terms of the ability to organize the vast volumes of data that Dr. B was talking about, and then the challenge of the analytics that are applied to that data to be able to look at it. All of the data in the world is pretty much useless if it lacks integrity, and integrity can best be controlled by software that allows us to monitor the input that's going into the system to make sure that it makes sense, to make sure that nonsensical values aren't entered. And we'd like to point out that not all the bioinformatics tools out there are created equally. So a suite of bioinformatics tools from a single provider simplifies the support and training because there's usually a commonality to the user interface. When we look at it from Abbott Informatics standpoint, we look at a few key elements that make up this suite of integrated applications that scientific enterprises use. One is the LIMS, the Laboratory Information Management System, that does the heavy muscle processing of samples, data entry, the communication with data integration systems and so forth. The scientific data management system that allows us to parse through documents and create some intelligence for how we look at data. Of course, the integration with instrumentation in the field. There is a requirement by some companies to be looking at cloud environments, and we certainly provide the ability to host in the cloud. These are largely attractive to companies that do not want to get into the IT business, do not want to be babysitting hardware devices and computers and networks and those kinds of things, the constant updating of software. There's the ever-present interest in mobility applications. Mobility, as we extend beyond your desktop computer to a laptop computer to tablets that can be highly mobile and walking around to different laboratory entities and laboratory locations and so forth, even down to the smartphone level and being able to use smartphones to do some low footprint or low screen area task. Advanced analytics, of course, is a way of consuming the data, presenting it, looking at it in different ways. That's become a whole cottage industry in the informatics field, and we partner with some of the key players in the advanced analytics field. And adding on to what Dr. B was talking about, as we start to talk about biorepositories, we do offer a biorepository solution, and I know that some folks in the crowd today are probably in the biorepository business. Others are looking at how to access that kind of information. We'll touch on all of that in just a few minutes. Essentially, we're talking about storage management, the management of capacity, the ability to annotate data and metadata, traceability, being able to monitor transactions. Searching and query, of course, is one of the main hallmarks of working with these kinds of applications. Track and chain of custody is not only required in many cases, but it's very nice to know where samples are in the whole process as they move around. Of course, diving a little bit deeper into the genealogy, spread coding, being able to issue work order requests and sample distribution are all key to biorepository functionality. In our world, from Avid Informatics, we've already implemented biorepository solutions with some of the providers that you see here. Not only that, but we've extended beyond the United States and internationally. We have serviced a lot of huge biorepositories around the world. So quite well accepted, quite functional, and quite robust functionality that we provide in this field. Looking at a few of the specific functions that sort of link into what Dr. B was talking about, query and ordering of specimens is a function of biorepository software. Without going into great detail, we have to, over the years, we've refined our interface to be making sure that those folks that are working in the laboratory, that we're not creating more work for them, that we're alleviating work. We're making it easy to find information, to tie it together, and to dive down deep into it. Specimens, especially when you're talking about thousands, tens of thousands, hundreds of thousands of specimens, they're not much good to anybody if you can't find them. So we've spent a great deal of time in organizing the hierarchy of our specimen storage functionality in our system. We get down to a great degree of granularity, the positioning of samples, not only the racks and the freezers they're located in, but the actual orientation within those racks. So we allow for great flexibility in structuring these containers that they're stored in. We take care of all sorts of shapes and dimensions and colors as well. This slide just refers to the Rutgers project, in which we have been servicing for several years, a very advanced biorepository. And I think the key points to take away from this slide, and this is a specific case study, are talking about the process improvements and the efficiency that we brought to Rutgers. It's been a very great partnership for Avid. Not only do they use a great deal of Avid instrumentation, but they do use our biorepository functionality and have been very successful in servicing their clients that use their biorepository. Genealogy tracking we touched on briefly. Of course, in this field, being able to dive down deep in the genealogy to see where certain specimens are linked to, their crossing of data into other specimens and so forth, all functionality that we've had to flesh out to be able to meet the needs of our customers. And of course, as we spoke about the mobility platform, the ability to look and scan barcodes with portable devices, both one-dimensional and 2D barcodes. You see one-dimensional barcodes here. We even allow for RFID tracking and so forth over the large scale of samples. So any means of identifying samples, tracking their movement through a laboratory is something very critical to any kind of a solution in the biorepository field. So we try to provide a lot of functionality attendant to that. Of course, reporting, developing reports, exotic or simple, is something very critical to this business. We try to make it very intuitive interface for developing reports. We allow the linkage to diseases and sample types of patient demographics, all this kind of information. We can assign rules to the templates so that they're very easy to set up. And we can also allow hyperlinks in forms of hypertags to be incorporated into the report. You can also draft the report in your conventional tools such as MS Word and of course crank them out into PDF documents. We use a strategy called fusion to tie together instrumentation in terms of acquiring data from instrumentation. We spoke briefly about RFID process that in itself warrants its own server. Where you see grabbers on this diagram refers to monitor processes that wake up and look for data to be coming in, whether from point-of-care devices or instrumentation, other databases, anything that is feeding into the system. So we try to provide a very fluid and robust instrument integration platform. One thing that we would like to talk about is that you should really understand your needs when considering bioinformatics as a platform. And as you look through the different providers, we should encourage you to seek a solution provider with a proven track record and a lot of deployments in this space. And if you think about the fact that there are many other tangential applications that apply to the bioinformatics field, we talked about analytics, we talked about the ability to provide mobility platforms and those kinds of things. We want to simplify life by teaming with a total solution provider, making sure that you have just one set of training, one set of user interfaces and so forth to be able to consider. And with that, we're ready to turn it over for questions, I believe.
Thank you, Randy. As a quick reminder to attendees, please take a moment to take our exit survey after the webinar has ended to give us your feedback. We will now enter the Q&A portion of the webinar. As a reminder to attendees, you can type in a question for the panelists at any time during the webinar by clicking on the Q&A box in the upper right side of the control panel. Our first question is for Atul Butte. You mentioned the opportunity in using various public and private databases containing existing data. However, I'm wondering about normalization. For example, for the GEO database, normalizing within a data set, between data sets, between arrays, and RNA-seq, etc. Can you comment on this?
Yeah, sure. So it's a great question about normalization. So indeed, a lot of these technologies have been used for many years now. RNA-seq is newer, and you have to be careful. So in general, though, that there's so many people have been running these types of measurements that you can really have faith in what you're finding in common. So why do I even believe in public data and the source kind of data here? The Microarray Quality Consortium over the past decade and a half has really shown that samples in general do measure up, no matter which type of RNA technology was used. I think they've recently done this now with RNA-seq, but certainly the microarray technologies going back to 2005. A lot of those papers influenced me because I knew that in general the measurements were there. So if the measurements are essentially there and they're actually accurate and they resemble each other, then it's just a matter of normalization. So there are many normalization methods that are out there. One of the participants already texted about lemma and some of these others. We tend to use crude kind of rank-based normalization methods. We've tested a bunch of normalization methods across diseases and tissues and use that to kind of influence our work. I'm sending out a link right now on the comment stream about a paper that we published back in 2010. 2009 on the normalization methods we use. So in general, they do work, and in general, the measurements do match regardless of the exact technology, at least at this blunt level that we're looking for. We're not looking at the subtleties here. We're looking for major differences here that are in common.
Thank you. The next question is also for Atul. Do you recommend using model organism data, or do you go directly to human data?
Yeah, so for developing our diagnostics, we tend to use whatever data is out there. So if people have model organisms and human samples, we actually try to put them into the mix. I'm not the kind of, I don't particularly believe in any one kind of religion like that, like humans are better than mice, in early stage research. In other work we published on type 2 diabetes and others, we tend to intersect across species and including human data just to figure out what's in common. Usually mouse models are at least resemble some aspects of the disease, and we'd like to capture those and intersect that with the human data as well. For preeclampsia, it's particularly harder because people might know there is really not a decent mouse model for preeclampsia, so there isn't a lot of model organism data, but many patients get it. So that's why there's a lot more human data than model organism data right now. And obviously to test the data, to test the samples, if you have access to the sample banks, whether commercial or your own, and they're organized, then there's no reason not to go straight to human samples to test them. Why? I mean, again, it's really more of a cost factor, but there's no reason why not want to go straight to that finish line.
Thank you. The next question is for Randy Heiss. Randy, we have many analysts and technicians who will only work with data entry screens. Can we limit the number of functions they have access to so their screens are not so confusing?
Yeah, one way that we approach that is to make life easier by assigning roles to different users. So a role might be limited to a certain set of screens or functionality, so their initial splash screen that they come into when they enter the product is just limited to the functions that they work with. So they don't have to sort through all these menu clicks and to try to get down to where they need to be working. So you can have multiple roles if you have both roles as a bench level analyst and also as a supervisor. You can have different sets of roles for those, so you see different functions, but it does tend to make the whole landscape a lot simpler to work with.
Thank you. The next question is for Atul Butte. Atul, where do you see deep learning and other artificial intelligence techniques to speed up drug discovery and disease cures?
Yeah, so a lot of these methods use very crude machine learning. Clearly, even just linear regression is the simplest form of machine learning. But machine learning is up and down the work that I'm showing you here. Sometimes, so a lot of people are using this phrase deep learning, and they use that to mean machine learning and artificial intelligence. To be kind of really strict about it, deep learning is one kind of machine learning where you're using particular multi-layer neural networks. Just to be instructive here for a second, when you look at this field of machine learning, there are two broad categories. There's unsupervised and supervised. Supervised means that you have all this data and you have an answer. You know, yes, this is cancer, this is not cancer, this is bad, this is good. You know what the right answer is, but you're trying to figure out an easier way to get to that answer. And so you can use the data to figure out, use this one and multiply by that one. You could come up with formulas. You can figure out a way that to use which features to tell what the right answer is. With supervised, you can measure accuracy, sensitivity, specificity. You can plot ROC curves. That's supervised. Unsupervised means you don't have a right answer. You have these samples. You have a lot of data. You're trying to figure out if there's intrinsic structure. Clustering, for example, is an example of unsupervised. And in some ways, deep learning could be used for both, but in general, most of the people are using deep learning for unsupervised type of research. For example, the famous researchers at Google put in all the YouTube videos they could, and because so many people upload videos of cats, they showed that one neuron in their multi-layer neural network, one neuron was just assigned for figuring out where the cats were because it just learned so many people uploaded cats. So something like that, you don't really have a right answer. You're trying a lot of different things and just seeing maybe something is interesting or not. So in general, in my own lab, my own approach, we go for supervised ones because we know a right answer. We want to get to this diagnosis faster, more accurately, cheaply. We know that we know what we're going after, so we try to figure out faster, more efficient ways to do it. And you find those using supervised techniques. You can use deep learning for that too, but so certainly machine learning is up and down this kind of informatics methodology. Did we lose our question asker here? I think we lost Ben, maybe.
Atul?
Yes, I'm here.
Oh, are you able to hear me?
Yeah, and I can now.
Thank you. The next question is also for you. Which statistical software do you recommend?
Yeah, so it's a great question. In general, so we use R and Python in my lab. We actually don't even standardize on either of those two. Most people come in with some expertise in one or both. But really, it's R and Python. Because they're free, they're scalable, I can get it to work in my on my laptop and then scale it up to our cluster or a supercomputer because you can get that to work anywhere. So in general, we use open source types of software packages. way back when, 15 years ago, I was using MATLAB. Never saw a problem with it, but I love the free and open source ways to do it. And just to expand on that answer, because someone else asked exactly what kind of computers, are we compute limited too? As people might know, I recently moved from Stanford to UCSF. I actually left the clusters running at Stanford. It wasn't even worth shutting them down. They were going to charge me to shut them down so they could go shut them down themselves. And my lab moved to Amazon. So we have one head node in our data center here. And for anything parallel-wise or large-scale computing, we just actually spin up nodes on Amazon to do it now. So we're not technically compute limited anymore because we're just renting computer time from Amazon.
Thank you. The next question is for Randy Heiss. Randy, how do you overcome the lack of standardization in the LIMS market?
Well, the lack of standardization, I believe, comes from the philosophical approaches of the different suppliers of information. How they actually program their products, whether they're truly web-based, what tools have been used as the underlying technology and so forth. It all tends to look the same to the casual observer, but underneath the covers, how it functions from an IT perspective is something that is of interest to those folks that are working that end of the spectrum. I think the other thing is that there has to be a consistent sort of interface to be able to acquire data from complex instrumentation and so forth. So from that respect, the acquisition and consumption of the data is somewhat standard because of the limits put on suppliers of these products in terms of data that is coming from the instrumentation. But once it gets into the LIMS system, then it's somewhat unstandardized. And so what we encourage folks to look at is, are the underlying tools something that are sustainable? Are they something that are public domain tools and so forth that are being used to construct the product? Or are they more linked to major software platform suppliers such as Microsoft and so forth? So I think that that's one way. You're never going to get around the different philosophical approaches of the different vendors, but making sure that there's consistency in their user interfaces and the underlying tool platforms is probably the most important consideration.
Thank you. The next question is for both panelists. What do you consider to be the biggest challenges for public big data?
Sure, I'll go first. So I think the public big data is incredible. I'm obviously a big fan of it. I think no matter what, even in this 1 hour, we are adding a couple hundred samples to Geo and all the other repositories. So we do have this culture now that we do have to share this data. The journals make you do it. The funding agencies make you do it. I think there's still challenges in knowing how to make everything seem easy, right? So learning how to download the data, process it, the statistical work, the tools. Most tools in this space are crude. It's great to get polished tools in bioinformatics when you can find them. I think that's always a pleasure to work with. But sometimes you have to deal with the more raw level software programming, things like that. The biggest challenge in this field is that there are not enough people who know how to use this data right now. I think there are a lot of biologists that know how to generate the data and maybe analyze theirs, but very few who download the data back and actually use other people's data. So I think that's to me is the biggest challenge in this field.
I think,
Randy, would you like to add anything?
Yeah, I just think from looking at the other end of it is looking at Dr. Butte had some interesting slides on the amount of data that's being passed around and available and so forth, and the quantities are so staggering. If you think about the storage technologies evolution over the past several decades, I think back when in the old days, my early part of my career with Digital Equipment Corporation, we had the removable disk drives that were very expensive. They stood 4 or 5 feet tall, and they contained a fraction of the amount of storage that the typical iPhone has today. So looking at the improvement in the storage technology, the ability to manage that data, that's been a challenge, but that's being met. I think the other thing is the reliability of networks. If you think about the whole questions that are attendant to people searching online for information, you look at the fact that, say, one of Microsoft's key initiatives is like Office 365, sort of cloud-based tools out there. that are sitting on a distant server. And 20 years ago, no one would have had the confidence that the networks would have been sustainable, would have been redundant enough, would have been robust enough to be able to pass that kind of data around and keep the connections active and so forth. Now it's just taken for granted because of the huge data centers that are serviced by folks such as Amazon, Google, Verizon, those kinds of things. The fact that you have the super redundancy of computing power, this incredible uptime for networks, 99.9% in some cases. It is, and also all of the security functions that are going into cloud-based platforms. The fact that most people were concerned that their data was insecure, now they're comfortable that the providers of cloud-based technology have really taken in data security as an extremely serious desire for the user base out there. So those are the challenges, but I see them all being addressed because it is the behavior of the consumer market that is pulling the suppliers into the direction that they need to be pulled.
Thank you. The next question is for Atul Butte. Atul, when developing diagnostics, what's the advantage between starting with data versus starting with samples?
That's a great question. I don't think there's a right way either way. I tend to be a data person, so it's easier for me to start with data. I think it really depends on your philosophy. If you think you really have the very best samples from the very best characterized patients, I think people tend to start with the samples. Of course, it depends on what kind of diagnostic you're trying to get to. There's still many new measurement platforms we're learning about constantly. You know, cell-free DNA, cell-free RNA, all these kinds of measurements are still new and might end up becoming great diagnostics. For example, non-invasive prenatal testing is all about circulating DNA. So there's still this idea that you can go after a new way of measuring things in a sample, and that might become a diagnostic. The data way is different. The data way is we've already had ways to measure molecules, and instead of starting with one experiment, start with 10 or 100 or 1,000 of them to figure out what's in common. So one could argue that it's more robust to start with more people's experiments than just the one that you're doing yourself, but that's really a hypothesis. We don't know for sure if that's really the case, but intuitively, to me, it seems like it is. So I think data people tend to start with data, sample people tend to start with samples. I think the goal in the end is to get to the most robust, most accurate, most sensitive type of diagnostic you can get to. And in the end, I think even if you start with samples, you're going to want to look at data too to figure out, does it match what others have seen or not? And if not, why not before you pursue it further?
Thank you. Next question is also for Atul. Outside of the legal pathway, how do you think labs could be incentivized to share their information, for instance, variants, to a centralized, publicly available database?
Yeah, that's great. So first of all, the legal side, there's very little legal here, okay? So the motivations to share come from all sorts of sources. The biggest one is really the journals, I think. When the reviewer's actually checking, they do require you to actually submit this data and have an accession number. The second are the funding agencies that typically don't award you money or actually don't renew your money, don't give you more money if you don't really make your papers open access and now your data. So the funding agencies, for example, DB-GAP, so if you're funded by NIH, I think Wellcome Trust and HHMI to do genetics work, you're supposed to deposit your raw data into DB-GAP or the equivalent. That's not, so that's a slight answer to your question. So, but people doing GWASs and things have to deposit data there. It's a little bit more burdensome to get data out of those repositories because of the patient re-identifiability problem, but you can still get data out of those. And then the legal part is different. I mean, legal is when there's actually a law that you have to share. So, for example, clinical trials data, you have to register trials in clinicaltrials.gov now. That's one example of a legality, an FDA regulation that makes you share some aspect of your trial and the results of that. So for variants, so we do in the research world, you have to share that data in DB-GAP. I see a bunch of genetics kind of people on the attendee list here, so you all know that. I think what the questioner is really asking is about clinical variants. So when I find a variant in the clinical world, in a clinical lab, what would incentivize me to share that or not? There are NIH repositories and consortia working on this. The ClinGen, ClinVar are working on this, trying to get the labs to submit their raw data. Of course, it's variable how much clinical data comes with that, so I think that's kind of a tricky part. But incentives really, I mean, really you hit the nail on the head by even using the term incentives. If there's a monetary aspect to it, if there's a reason why people would feel like they're getting paid for this or there's some reward for this, I think you would incentivize a lot. Asking people to do it out of the goodness of their free will, I think you get some, but you're not going to get all of them. So I think you can create incentives, whether it's monetary, whether it's federal government giving grants for this type of work. I think those would be ways to do it from the clinical side.
Thank you. The next question is for Randy Heiss. Randy, barcoding is a huge part of our operation because we have thousands of specimens. Some of these specimens are stored in harsh conditions. Can we use barcodes that stand up to very cold temperatures?
Yeah, so the providers of barcode technology have so improved over the past decade or so. Not only, as we addressed, do they have tiny, affordable RFID stuff, which is pretty much impervious to some of these harsh conditions, but even the materials that are used in barcoding labels can be stored in super frigid temperatures without chipping or coming off. That's their living. So there's these specialists in those fields that have created these materials to create barcodes that can resist solvents, they can resist cold temperatures, hot temperatures, you know, stability, chamber conditions, those kinds of things. And so I think that there's no worries there. There's such competition among those. The important thing is, from a software perspective, that they have an easy, clean interface that we can talk to. And since that's their living, they make that very easy for us to do. So I don't think that there are any problems out there that I've seen that can't be solved by some of these specialized vendors of barcoding systems.
Thank you. The next question is for Atul Butte. Atul, can you provide some general strategies for using downloaded metadata?
That's great. So downloaded metadata. So obviously these repositories have the data in various different forms. For example, for most of the experiments I showed you are from NCBI Geo. There's a lot of text-based data, some structured data. There are packages that you can use that feed into databases to help organize that. So there's RGeo. I run a repository for National Institute for Allergy and Infectious Disease called Import, where we have clinical trials data, immunology data, flow cytometry data. We have a package in Bioconductor called R-Import. So their data structure is there. That doesn't really answer the question about databases, though. So in the end, you kind of have to build your own database to hold the data, depending on how much you want to download. We've come up with really crude solutions internally. We tend to download a lot of public data and put it into one table space. As a bioinformatics lab, it makes a lot of sense because then you can do all those joins, what we call table joins. You know, I love it when a postdoc comes in and says, well, yeah, I see something in common between this genetic data and this microarray data, and they can, because it's in the same table space, they can start to build queries to try to intersect all that. And it intersects with dbSNP and NCBI gene and get the symbols and the names of these genes. So the idea, going back in my lab, was to put it into one table space. There's no established way to do it. I think there are a bunch of papers that came out about 10, 15 years ago about how to organize these databases. Now I get the sense that people do it and don't even write papers about it. These are actually tough papers to publish. There are companies that are trying to organize this. I remember the Next Bio was trying to do this, I think, for companies like a subscription kind of thing to organize your own data. But again, you have to be a paying customer to do it. I think there's probably others now that offer commercial solutions to organizing your own molecular data and presumably by extension probably some of the public data as well.
Thank you. The next question is for Randy Heiss. Randy, we have several sites with similar laboratory workflows, but they currently use tools that are much different than ours. How can we arrive at a common solution?
Yeah, the very best answer there is you have to abstract the look at the workflow from the individual sites, because the folks that are working at a particular site are so involved in their day-to-day activities that they don't have time to look at how their other sites within the company are doing their activities, and so there's usually no need driving harmonization across multiple sites. If there is a group that's capable of doing that, or if there's a need to look at a consulting body to come in it makes a lot of sense because with the advent of web-based applications, you can have a centrally served system. But if you're going to have a centrally served system, that necessarily implies that the workflows have to be harmonized and normalized across several sites. So sometimes it's a little easier for an external consulting body to come in and help with that because they're not viewed as having any kind of parochial bias, that they will be open to ideas that improve the overall greater good of the company from a business perspective. And it really tends to open the eyes of folks if they look at their workflows in kind of a joint effort because they get to discover each other's operations in greater detail. And then by abstracting it back to the basic business need, you're able to divorce from the kind of site-specific bias and that they've been doing things a certain way for many years and look at improving the process from an overall perspective. So I guess the short answer is if there are folks inside of the company that understand how to look at and diagram workflow across sites, usually business analysts in the IT department or so forth, they're good to get involved with that. But if there's some sort of friction between sites because of the way that they do things, there are a number of consulting bodies out there that can help bring these kinds of activities together in a kind of a neutral format so there's nobody with hurt feelings.
Thank you. This concludes the Q&A portion of the webinar. We'd once again like to thank our panelists, Atul Butte, Director of the Institute for Computational Health Sciences and Professor of Pediatrics at the University of California, San Francisco, and Executive Director for Clinical Informatics at the University of California Health Sciences and Services, and Randy Heiss, Manager of Global Strategy for Abbott Informatics. If we didn't have time to get to your questions, we will try to have them answer directly afterward. As a reminder, please take a moment to complete the brief survey that should pop up on your screen before logging off from the webinar. Your feedback is extremely valuable. If you missed any part of this webinar or wish to listen to it again, a link to an archived version will be emailed to attendees. Thank you for joining us for this genome webinar.